Namaskar, my dear students. Today we are going to start with the new unit of the botany portion and the very important chapter, and the name of the chapter is the genetics. In genetics, we shall be dealing with the various aspects of genetics, including the principles of inheritance and variations, molecular basis of inheritance, and so on. Now, before I start with this chapter, I would like to ask you a few things. That have you ever made this simple observation that why all the individuals, they have a definite individuality. What I mean to say, that one individual, one human being is easily distinguishable from other in variety of ways. Another thing that you might have noticed, that dogs, they are of the several varieties, and even one dog or the other plant or a cat or a tree, that is distinguishable from the other, even they are of the same breed or the species. Another point of interest is that when individuals have offsprings, they produce the individuals of their own kinds. That is, dog always produces a dog, never cats or rats. Furthermore, we know that babies of two white skinned people will be white and not black skinned. So these are the certain mysteries of the nature and miracles of the nature that are unfolded in the genetics. Now, let me introduce you with the topic, what is genetics? The branch of biology which deals with precise understanding of the biological properties which are transmitted from parents to offsprings is called genetics. The word genetics is derived from the Greek word genesis, which means to grow or to become. We have heard many times a statement that a son has mother's eyes or a father's nose. These similarities are due to the inherited characters or the heredity. And now heredity is the similarity of the characters between one generation and their offsprings. Now this term genetics, it was proposed by William Bateson, W. Bateson, in the year 1905, when he gave the definition that process of transmission of characters from one generation to another is known as inheritance or heredity. However, it would be mandatory to mention here that father of genetics is Gregor John Mendel. He is considered the, no doubt the term genetics was given by William Bateson, but Gregor John Mendel is the father of genetics. And next, as I told you that every individual has an identity, so it means there are certain similarities between the parents and the offsprings, but of course there are some variations between the offsprings and the parents, and there are these differences are called as the variation. The differences shown by the individuals of a species and also by the offsprings of the same parents, that is brother sister, two brothers, two sisters, they are the variations, and these differences are referred to as the variations. To be a little in more detail, I would tell you that there are the variations are classified into two ways, according to the nature, according to the degree. According to the nature of the variations, they are of two types, somatic variations and the germinal variations. Now, what is this somatic variation? Such variations are not inherited from parents and affect the somatic cells only. As dear students, you know there are two types of cells, somatic cell and the germinal cells. The somatic variations come in the somatic cells only. They are acquired by the organisms during its own lifetime, and they are lost with the death of the individuals, and hence Such variations are also called as the acquired variations, since they are acquired during the lifetime. This may be due to the environmental factors, due to nutritional variations, due to variation in the habitat, due to better conditions or the poor conditions of existence, due to use and disuse of organ, or due to some conscious efforts that we shall be discussing in detail uh, afterwards. And then on the basis of degree, variations could be continuous, they could be discontinuous. Now what is this continuous and discontinuous? You must understand it. Such variations, the continuous variations, are those variations which are small and indistinct when compared to the normal. It's found in both on the plus side as well as on the minus side of the normal type. These are also called as the fluctuating variations, and these are unstable and non-heritable. The continuous variations could be substantive, could be meristic. That is, variations which bring uh, changes in weight, size, etc. They are called as the substantive, and which gets a change in the smaller things such as in the number. They are called as the meristic, such as the number of arms in the starfish, number of segments in the earthworms. These are the meristic variations. Now, what is discontinuous variation? Such variations are large and represent the conspicuous differences in the progeny from the parents, and they are called as the mutations or sports or saltations. They appear suddenly, they are stable and are inheritable. Now, continuous variations, these are also the variations which are non-inheritable and unstable, whereas the discontinuous variations, they could again be substantive, they could again be meristic. The substantive mean variations in the size, height, weight, shape, color, etc. Meristic means in number. For example, polydactyly. Polydactyly is what? When there you might have seen some persons who have more than 5 fingers, that is 6 fingers in their hands, that is polydactyly, and that's a meristic change. So, could you ever think what is the significance of these variations, dear students? They constitute the raw material for the evolution. By introduction of variation, useful variants of plants and animals could be produced. They could be the basis of heredity. They might help in the adaptations, as the environment is also ever changing, it's dynamic, and so the individuals are also dynamic, and small changes, they might bring about the adaptations. Variations make some individuals better suited in the struggle for existence, and variations provide such organism a distinct individuality. So variations as well as inheritance both together play a role in bringing about the evolution, in bringing about the new species, and in fact making possible the humanity. Now I must mention here, it's mandatory to tell you that the modern concepts of genetics, heredity, variations that today we have, they are not the result of a single step. They have evolved, and it has always been a term of curiosity for the mankind to find out why parents and offsprings resemble, why some variations, and accordingly from the time to time, according to the resources, according to the development, according to the discoveries, the views were given by various thinkers, philosophers, scientists, etc. And some earlier views are mandatory to be discussed here so that we can reach the modern concepts of the inheritance or the genetics. The first view, the some main points, some viewpoints were put forth before Mendel to explain the transmission of the characters, and one such theory was the moist vapor theory, which was given by a Greek philosopher Pythagoras in about 580 to 500 BC. who proposed that each organ of the animal body produced vapors and new organism was formed by combination of different organs. Might seem to be ridiculous in the today's scenario, but it was the theory which and the curiosity which led to the modern development of genetics. Another theory was the theory of performation. Leeuwenhoek was the first scientist to observe the human sperm, but this theory was given by Swammerdam, a Dutch scientist. This theory believes that one of the sex cells or the gametes, either sperm or egg, contained within itself the entire organism in perfect miniature form. That is preformationism, and the theory is the preformation. Miniature form was called as homunculus. The theory was supported by Malpighi, etc., and Rox, one of the main thinker of that time. And this theory was again discarded since it had no basis of its own kind. Then came the theory of epigenesis, which was proposed by Wolff in 1738 to 1794, who proposed that many such factors such as tissues, organs appeared during the development of one organism which were not present in the original form. So this was called as the theory of pangenesis. Another very interesting theory was the theory of last one was the theory of epigenesis. Now theory of the pangenesis, given by Charles Darwin, the founder of modern evolutionary theory. He believed and proposed his views in 1868 in the light of the cell theory and knowledge of embryology that very small, exact but invisible copies of each body organ component, to whom he called gamules, were transported by bloodstream to the sex organs and were assembled into the gametes. Upon fertilization, gamules of the opposite sex were added. This young one has a blend or the mixture of the pangenes, hence with the characters of both the parents. The theory implied that changes to the body during an organism's life would be inherited as proposed by Lamarckism. The Lamarckism we shall be discussing in the coming lectures. Now in the figure you can see the body is forming the egg and the gamules from different organs are coming to the egg, leading to the development of embryo and hence the entire organism. And several other theories, they were still coming in the picture till the great discovery, the original discovery, which was the founder discovery, was given by Gregor John Mendel. Now let me discuss, it is called as the modern genetics. Now let me discuss something about the great scientist, the father of genetics, Gregor John Mendel. Gregor John Mendel was born in the year 1822 to a family of farmers in Silesian, a village in Henserdorf, which is now a part of Czechoslovakia. He finished his high school at the age of 18. He got this early education in the monastery in Brno, previously Brunn of Austria. At very early age, he was ordinated a priest of Augustinian monastery at Brno. A few years after, he was sent to University of Vienna in Austria for training in physics, mathematics and natural science. It was at Vienna that Mendel was influenced by two scientists, Franz Unger, a plant physiologist, and Christian Doppler. The Doppler you might have heard. He is discoverer of well-known Doppler's effect in physics. He was very much influenced by these two scientists, and Mendel picked up the knowledge here of the hybridization experiments, and in 1854, I am talking about the legacy of the Gregor John Mendel. In 1854, he continued to work as a priest and as a teacher in high school. In 1857, he began his famous experiments in peace in the monastery garden, and he carried out his experiments for 7 years from 1856 to 1863. And he worked on the uh, theory of heredity and or the inheritance, and he presented his results in the form of a paper, and the paper was entitled as Experiments in Plant Hybridization, which was read before Brunn Natural History Society in 1865. But his discoveries were unnoticed because people thought it was an obscure journal. Scientists failed to notice that his work because world was busy in controversy of the Darwin's theory of origin of a species. No, moreover, no solid findings he could give in the absence of the modern discoveries of the electron microscopes, etc. His ideas were ahead of his time, as the ignorance was prevalent in that about cytological basis of heredity. In 1900, his experiments were rediscovered by three scientists, Hugo de Varis, Karl Correns, and Teschermark of Austria, and then he got recognition and that was the time of 16 years after his death, and now he is called the father of genetics. Now, what he did? Today we know that genetics is a field of biology that shows the characteristics or the traits which are passed from the parents to the child. And because he laid the foundation of this phenomena or the theory, he is called the father of genetics. And he took pea plant Pisum sativum as an experimental material, and he selected 7 characters in it: its height, pod appearance, seed texture, position of flower, flower color, pod color, and seed color. And uh, now he was advantageous in bringing about his discovery because pea plant was a advantageous plant. And what were the advantages of selecting the pea plant for the experiments by Mendel were that pea plant had a shorter period of time and several generations could be studied in shorter period of time. It was easy to grow and needed little care. They show several pairs of visible characters with contrasting traits because inheritance also occurs of some characters which are not apparently visible, but he selected apparently visible character. Pea plant reproduce sexually and by self-pollination, so hybridization experiments were easier and could be well understood. Now, the 7 characters that Mendel selected for his experimental material, they were showing two contrasting traits. One trait was called as the dominant character, other as was called as the recessive character. For example, in seed shape, it was round dominant and wrinkled seed was the recessive character. Seed color, yellow dominant and green recessive. Seed coat color, it could be colored it as a dominant character and white as a recessive character. Pod shape, inflated and constricted, dominant and recessive respectively. Pod color, green and yellow. If it was green, it was dominant, yellow, it was recessive. Flower position, axial or terminal. Axial was dominant character, and if it was terminal, that is at the end of the branch, it was recessive character. Stem length, it could be long, it could be short. Short was recessive and long was the dominant character. Now before I proceed with the experiments of Mendel, I would like to introduce you with some terms. For example, two terms we are talking here, dominant and recessive. We must understand what are these two terms before we actually proceed with the Mendel's characters. and these terms will be called as genetics terminology. What is dominant? Trait which stays visible. That is, if a child is born to a parents of who are having, mother is having curly hair and father is having straight hair, and a child is born with the curly hair, it means the visible trait is the curly hair, and that is the dominant trait, and trait which has disappeared is called the recessive trait. Another term is the allele, that is alternate forms of the genes. For example, curly and straight are the two alternate forms of the single gene. To what we call gene today, Mendel called it factor. Genotype, genetic makeup of particular trait, and phenotype means what is visible, that is physical appearance of the trait. Two more terms, homozygous and heterozygous. Homozygous means when both the alleles are of the same type, either whether dominant or recessive but same, then it is called as homozygous, and heterozygous when two alleles are of the different types. If you understand these terms, we are in a position to move to the further, and I would like to tell you here that Mendel proposed 4 laws of genetics on the basis of 2 crosses he made, and the 2 crosses were the monohybrid cross and the dihybrid cross. Now what is monohybrid cross? A cross between a pea plant that is homozygous for yellow and a pea plant homozygous for seed will produce only yellow heterozygous offspring. What I mean to say by this, when we take into consideration only a single character, because when a offspring is born, it carries several characters from parents to offsprings, some in dominant traits, some in mixed traits, some in recessive trait. But when we under our study we take only one character, that is called as the monohybrid cross. On the other hand, a dihybrid cross is a way to predict the genotype and the phenotype of the offspring when we take two characters into consideration. For example, color and size, position and color. That is when we take two characters into consideration in a particular experiment which we are performing, that is called as the dihybrid cross. These were the 2 crosses, the monohybrid cross and the dihybrid cross, which were undertaken by Gregor John Mendel for his experimental material, and he gave 4 laws of inheritance. We shall be discussing these 4 laws of inheritance, Mendelian concepts and the post-Mendelian concepts in our next lecture. Till then, thank you. Stay safe and please go through this lecture thoroughly so that you can understand the subsequent lectures. Thank you.